Hello everyone, this is Dr. Satish S. Bornare, Department of Genetics and Plant Breeding, KK College of Agriculture, Nasik. Today we are going to learn the topic breeding for insect resistance. So friends, this likewise diseases, the insects are important causal factors for biotic stress in crop plants. Means besides these diseases, the insects are also major one to cause the stress in crop plants. These insects attack all the crop plants and lead to considerable losses in yield as well as quality of crop plants. So these insects are harmful, and this insect attack leads to various types of damages in crop plants, such as reduction in plant growth or stunting, then damage of vegetative and reproductive part also, and premature defoliation also occurs due to the insect attack, and in major case wilting of the plants is also observed due to the insects. These insects cause roughly 14% estimated yield losses of all the important crops on global basis. So these insects are having economic importance and causing huge losses to our crop plants, either by feeding or sucking cell sap and eating away a various plant part like tissue feedings or cell feeding. And these insects also causes yield losses through the transmission of the various diseases, that is it acts as a vectors in different diseases also. So for control of these insects there are two important methods. First one is a biological method and second one is a chemical method. So these are the two important methods to control the insects, in which biological method, in this method the insects are controlled by three ways. First, by using the predators and parasites, that they are natural enemies of this insect space, and by using these predator and parasites we can control the insects. Then second, by using the botanical pesticides such as neem, datura, and various uh, formulations are available in the market. So by using these botanical pesticides we can control the insects. Then third one is by using the resistant varieties. So these are the biological method to control the insects. Then second one is method is chemical method. This chemical method includes use of various chemical insecticides with several as we all know that there are several chemicals available in the market, and these different insecticides are used by different farmers or in different farmers in different crops to control the insects. But as compared to this biological method, this chemical method is disadvantageous as it increase the cost of cultivation, reducing the population of the predators and parasites, which are the beneficial uh, natural enemies of our insect pest. Then this chemical method or using of chemical insecticide leading to environmental pollutions and development of pest resistance biotype of the insects. So these are the disadvantage of chemical method. So for that, to avoid the disadvantage of the chemical method, the genetic resistance is the cheapest and best method for insect control in crop plants. So what is the insect resistance or genetic resistance? These are the ability of some genotypes to give higher yields of a good quality than the susceptible varieties at the same level of insect attack under similar environmental conditions. Means here the genetic potential is used to confer the resistance against the different insects. Then what are the different mechanisms of the insect resistance? First one is a non-preference, then antibiosis, then tolerance, then avoidance. So friends, these are the four different mechanisms of insect resistance, and these mechanisms were given by a painter in 1951. Let us see one by one. First one is a non-preference. So this non-preference refers to the various features of the host plant that make the host undesirable or unattractive to the insects for food, shelter or reproduction. And this type of insect resistance is also known as non-acceptance or antigenosis. By word non-preference, we have an idea about the terminology. Means in case of non-preference, the host plant is such unattractive or undesirable characters which are non-preferred by the insects, and the insects will not accept a resistant host plant even if there is no alternative source of food. And the plant character associated with these non-preference includes color, light penetration, hairiness, leaf angle, and odor and taste of these host plants. So these are the some important characters which are associated with or confers the non-preference to the insects. In case of here, some of the characters or some of the attributes of the host plant are given which gives the non-preference. First one is in case of cotton bollworm, the red plant body, smooth leaves, long pedicel, then open canopy, nectarlessness, thickness and hardness of the bold rind. These are the some of the traits or attributes of the host plants that confer the insect resistance and prove the non-preference to the insects. Then in case of pea aphid, the yellow-green genotypes are non-preferable by these pea aphids. In case of soybean, the hairy genotypes are non-preferable by this soybean leaf hopper, and in case of cotton, the hairiness of the leaf and stem is non-preferable to the cotton jacids. So these are the some of the examples or attributes which confer the insect resistance by the mechanism of non-preference. Then second mechanism is antibiosis. This antibiosis refers to the adverse effect of host plant on the development and reproduction of the insect pest which feed on the resistance plant. Means once the insect feed on that plant, it is having an adverse effect on the insects, and resistance plant retard the growth and rate of reproduction of the insect plants, and in some cases there is a chances of the death of the insect in case of antibiosis, and thus this antibiosis is considered as a two form of resistance to the insect pest. And in case of cotton, the cotton antibiosis is associated with high level of this gossypol, then tannin, then halosides and silica content. Means if these compounds are in a higher concentration, which gives the antibiosis for different insects in case of cotton. Then next one is a tolerance. This tolerance refers to ability of a variety to produce a greater yield than susceptible variety at the same level of insect attack. This definition of tolerance was given by Maxwell and co-workers in 1972. So, according to the Maxwell and co-workers, this tolerance is the ability of a variety to produce a greater yield than susceptible variety. In other words, we can say a tolerant variety will give us a higher yield than susceptible one despite insect attack. And here the tolerance of the variety play a role. The tolerance is measured in terms of rejuvenation potential, healthy leaf growth, flowering compensation potential, and superior plant vigor. So these are the some of the terminologies or some of the terms in which we can measure the tolerance of the particular variety to a particular insects. And these tolerant cultivars have a greater recovery of a damaged parts than susceptible ones. So this is the another mechanism which confer insect resistance. And the last one is avoidance. This avoidance refers to escape of a variety from insect attack either due to the earliness or its cultivation in the season where insect population is very low. This is the simple mechanism by which the main season of an insect pest attack is avoided. For example, early maturing cotton varieties escape pink bollworm infestation which occurs late in the season. That means the early maturing variety of cotton does not have infestation of the pink bollworm because the pink bollworm observed in late season. So in case of that variety, early maturing variety of cotton easily avoid or escape the insect attack. And this avoidance is also an effective means to protecting crop from the damage of insect pest. So these are the four different mechanisms of insect resistance. Then next, what are the basis of insect resistance? So there are three important bases of insect resistance, out of which first one is a morphological basis or morphological factors, in which the different morphological traits such as hairiness, color of the host plant, and toughness of the tissue, these morphological traits confer insect resistance in different crop plants. For example, resistance to acids in cotton, aphids in turnip, and the leaf beetle in cereals is associated with these hairiness of leaves and stem. So this hairiness confers the resistance to the different insect or different insects in a different crops. Then color of the plant. This plant color contributes to the non-preference in some cases. For example, red cabbage and red leaves Brussels sprouts are less favored than green types. So here color is the important one. The red color is less favored as compared to the green one by butterflies and other lepidopteran insects for their oviposition in case of cabbage and Brussels sprout. Or in case of cotton, the red plants are less preferred than green by bollworm. So this color of the host plant, these morphological traits or features confers the insect resistance in different crop. Then sometimes the solid stem or toughness of the tissues also play an important role. For instance, in case of wheat, the solid stem confers a resistance to the stem supply, while the tough and thick plant tissues causes mechanical obstruction to feeding and oviposition of the insect pest. Thickness of the leaf lamina in cotton contributes jacid resistance. And similarly, in case of cotton also, balls with thick and tough ball rind are difficult for puncturing by bollworm. So these are the some morphological traits or factors which confers in insect resistance in the different crop plants. Then second one is a physiological factors. These physiological factors such as osmotic concentration of the cell sap and exudates or gummy exudates from the host plant. These two physiological characters or factors are also associated with insect resistance. For example, in some species of Solanum, the gummy secretes or gummy exudates from the Solanus species from the hair on the leaves, and aphid and collared beetle get trapped in such a exudates and are unable to feed and reproduce. So these gummy exudates or secrets of the solanaceous crops from hairs on the leaves confers the insect resistance in this crop. And several species of Solanum also or tobacco varieties secretes the exudates which are toxic to various insects such as mites and thus confers the insect resistance. And also osmotic concentration of the cells of the host plant is play an important role in conferring the insect resistance in the different plant. So these are the different physiological traits or factors which confers the insect resistance. Then third one is biochemical factors. So friends, these biochemical factors are considered as more important than morphological and physiological factors for the insect resistance. And there are number of examples of biochemical factors or traits are conferring the insect resistance in different plants. Let us see one by one. High concentration of saponin in case of the alfalfa in leaves and stem confers the insect resistance to the spotted bollworm and alfalfa aphids. So when this biochemical compound or factors is present in high concentration which confers the insect resistance against the spotted bollworm and alfalfa aphids in case of alfalfa crop. Then high concentration of gossypol. So this gossypol is a phenol component present in the cotton and which confers resistance to several insect pests in cotton. Then high concentration of benzyl alcohol. So this benzyl alcohol is majorly present in wheat and barley and which confers resistance to the green bug in case of wheat and barley. Then high silica content. This high silica content in case of rice in the shoot confers the resistance to the stem borer, and feeding of the insects on high silica content varieties leads to rapid wearing of the mandibular mouth parts of the insect. So this high silica content in the stem prevent the insect feeding or become a major hurdle for the insect for feeding. Then high concentration of this dimboe in maize. If this concentration of dimboe this showed higher, then there is a resistance to the European corn borer which is a major pest of the maize, and this biochemical compound gives the resistance against that European corn borer. And last one is a low concentration of the asparagines. These asparagines, when present in a low concentration in a rice, gives a resistance to the brown plant hopper. So these are the different biochemical factors which confers the insect resistance in different crop plants. Then genetics of the insect resistance. So friends, this insect resistance is governed by oligogenes, polygenes and cytoplasmic genes also. Here the oligogenes governed by oligogenes. Oligogenes means the resistance is governed by one or few genes, mostly one or two. For example, in case of wheat, to the Hessian fly and stem saw fly, and in case of barley, these green bugs, and in case of alfalfa, the pea aphids, and in case of cotton, the jassids. So these are the some of the examples of oligogenic insect resistance. Then polygenic insect resistance. This poly means many. So this, as name indicate, the resistance is governed by several genes and each having a small and additive effect by the definition. And for example, in case of wheat, the cereal leaf beetle, and in case of rice, stem borer, and in case of maize, earworm and leaf aphids. So these are the examples of polygenic insect resistance. Then last one is a cytoplasmic genes. As we all know that besides the nucleus, there are some cytoplasmic genes in the form of mitochondria or the cytoplasm. Mitochondria or ctDNA or mpDNA. These cytoplasmic genes, this resistance is governed by these cytoplasmic genes. For example, in case of maize, the European corn borer, and in case of lettuce, the root aphids. These are the examples of the cytoplasmic insect resistance. Then, what are the different sources of the insect resistance? So, there are 5 sources of the insect resistance. First one is a cultivated varieties. In some crops, genes for the resistance may be found in normal cultivated varieties, that is cultivar. For example, in case of cotton, varieties like SRT1, DHY 286, PKV 081 and B 1007 are the good sources for jacet resistance. So these are the cultivars or cultivated varieties which act as a source of insect resistance. And if the cultivated varieties, the desired insect resistance is not found, then the wild relatives or wild species are the potential source for the insect resistance, as these wild species are the home for the insect resistance and disease resistance also. Then germplasm collections. The germplasms or genetic resource are a good source of insect resistance, and these resistance lines are identified by screening of germplasm for specific insect. This screening is done when resistance is not found in cultivated species, and these germplasm collections or plant genetic resource play an important role in forming or the insect resistance. Then induced mutation. So this induced mutation is also important source for the insect resistance. For example, in case of strip rust resistance in wheat, lip spot and stem rust resistance in groundnut, and red rot resistance in the sugarcane, these all the insect resistance are obtained through induced mutation. Then last one is unrelated organisms. For example, in case of cotton, we all know that the Bacillus thuringiensis, the gene from that Bacillus thuringiensis or BT gene is inserted in the cotton through the genetic engineering, and using these unrelated organisms which gives the insect resistance. So these are the different sources of insect resistance. Then next is breeding methods that are used for insect resistance, and in this the introduction, this is the best source or best method for conferring the insect resistance. We can introduce variety from throughout the India or from exotic also and confer the insect resistance. Then second one is selection. This selection includes pure line, mass selection and different types of selection according to the mode of pollination of the particular crop and produce insect resistance variety. Then we can also breed the insect resistance variety by the method of hybridization, by combining the different gene source. And next one is a mutation. By using the artificial or induced mutation, we can create the insect resistance varieties in different crop. And by using transgenic breeding also, we can create the insect resistance. And bt cotton is the example, or there are many number of example in different crops where the transgenic breeding is used for insect resistance. So these are the some of the breeding methods which were commonly used in insect resistance. Then there are screening. There are two types of screening. First one is field screening and second one is a glass house screening. So the screening for insect resistance is done under both condition, under field or in a glass house. When there is a field screening, it is not possible to ensure uniform initial infestation of all the plants in the population, which is prerequisite for the screening. And if we screen the varieties under the field, then we have to take precautions, or we have to apply some measures so that the uniform infestation by an insect pest in the field are maintained. For that, the interplanting a row of a non-susceptible variety between two rows of the testing material is important one. Then screening is in highly prone areas. Then in case of soil insect pest to be tested, so it should be tested in a sick plots only. Then testing in a particular season when infestation is very high, so that the uniform infestation will be takes place. For example, in case of rice stem borer in off season, then transferring manually equal number of eggs or larvae to each test plant. So this encourage the uniform infestation by an insect pest, so that field screening will be effective. In case of glass house screening, results from glass house tests are much more reliable than those from field test, since both the environment and the initial level of infestation are more or less uniform for all the plant being tested. Here is the merits of the glass house screening. Then what are the different problems encountered in the breeding for insect resistance? First one, breeding for the insect resistance may lead to a susceptibility to the another pest. While we are tackling with the one pest, there is a chances of the infestation or susceptibility to the other pest, or it may be happen. So this is the limitations or major problem encounter in the insect resistance. Second one is a reduction in quality or make unfit for consumption. Sometimes it may happen that while we dealing with the insect pest resistance, the quality of the crop is reduced, or sometimes it may found unfit for the consumption. And third major one is there is a linkage between desirable and undesirable genes, and interspecific varieties are generally low yielding and their produce is often of inferior quality. So this is the major hurdle. As we all know that there is always linkage between desirable and undesirable genes. When we want to transfer the desirable one, there is a linkage drag of undesirable ones, and breeder have the major hurdle of these undesirable genes. So we can break this linkage drag. Fourth one is a screening for insect resistance is the most critical and difficult step in a breeding program. It necessitates close coordination among the scientists belonging to different disciplines. So friends, this screening for insect resistance is a multidisciplinary approach, and there is a necessary for close coordination among the different scientists of the different field. So can the breeding for insect resistance will be rewarding. Then last one is it is a long term and expensive program. This is a breeding for insect resistance is a long term and expensive method. So these are the some of problems or demerits of breeding for insect resistance. Here the practical achievements out of these, the different insect resistance varieties have been developed in many crops. Here are the some of the examples. In USA, the Hessian fly and stem sawfly were the serious insect pest of the wheat, and more than 23 varieties have been developed so far, and these varieties are successfully grown in Hessian fly prone areas of USA. This is the major achievements of the breeding for insect resistance. Then stem sawfly resistance variety rescue with a solid stem was released from the Canada in 1946, and this variety is grown successfully in sawfly prone areas of USA and Canada also. In maize, resistance varieties to rust have been released in maize, and rust is no longer serious disease. So this is the another major one important practical achievement of the insect resistance breeding. And same in USA, the barley variety will resistance to green bug has been developed potentially by USA, and cultivation of these varieties reduce the population of the insects in a greenhouse to up to 50%. Then alfalfa variety Cody, Mopa and Gia developed in USA are resistant to the spotted alfalfa aphid, and the oat variety Greta developed by Belgium is resistant to the stem eelworm. So these are the all practical achievements in abroad. In India also, the cotton varieties like B1007, SRT1, Khandwa 2, DHY 286, PKV 091. These varieties are evolved in India are resistant to the acids in case of cotton, and these varieties are successfully grown in the major growing area of cotton, that is central India. And cotton varieties like Kanchana, Supriya and LK86, 861 are the tolerant to the white fly. So these are the some of the practical achievements of the breeding for insect resistance. So thank you friends, this is for education purpose only, and references are as below. Thanks once again.